everyone. This is Kanchana from 4th semester Microbiology department. And good evening, sir. And my topic is about enzymes involved in degradation of lignocellulose. So I'll be covering on these topics. So what is cellulose? Structure? What are cellulose? Microorganisms involved in cellulose degradation, enzymes degradation, factors, mechanisms. So what is cellulose? So this is the structure of cellulose. Cellulose is an organic polysaccharide composed of a linear chain of 100 of beta-linked D-glucose units. So cellulose is the most abundant extracellular structural polysaccharide or organic polymer of all biomolecules in the biosphere. Cellulose is present in all land plants but is completely lacking in meat, egg, fish and milk. It is however not metabolized by the human system. It is the most widely distributed carbohydrate of the plant kingdom that comprises about 50% of all the carbon in vegetation. Cellulose occurs in the cell walls of plants where it contributes in a major way to the structure of the organisms. All cellulose synthesizing organisms include bacteria, algae, tunicates, and higher plants have cellulose synthase proteins which catalyze the polymerization of glucan chain. Even if the human body cannot digest cellulose, it acts as a source of fiber. In nature, cellulose is a source of food to a wide variety of organisms, including bacteria, fungi, plants, and protists, as well as a wide range of invertebrate animals like insects, crustaceans, annelids, mollusks, and nematodes. So I have taken one more structure of cellulose, this thing. Okay. The molecular weight of cellulose ranges between 200,000 to 2 million daltons. Cellulose consists of a D-glucose unit at one end with the C4OH group as the non-reducing end, and the terminating group is C1OH as the reducing end. The bond is formed by taking out a molecule of water from the glycosidic OH group on carbon atom 1 of 1 beta D-glucose molecule and the alcoholic OH group on carbon atom 4 of the adjacent beta D-glucose molecule. And hydrocellulose is the repeating unit of a cellulose. The overall structure of cellulose is a result of the binding of adjacent cellulose chains and sheets by hydrogen bond and van der Waals force, resulting in a parallel alignment. The structure of cellulose resembling in structure with amylose, except that the glucose units are linked in together by beta-1,4 glucoside linkage. What are celluloses? Celluloses refers to a group of enzymes that catalyze the breakdown of cellulose to form oligosaccharide, cellulobiose, and glucose. These enzymes represent a class of enzymes that are produced by fungi and bacteria that assist in the hydrolysis of cellulose. Cellulose is an important group of enzymes that play an essential role both in industries as well as in nature. In nature, celluloses are involved in the global carbon cycle by degrading insoluble cellulose into soluble forms. The complete enzymatic system of cellulose consists of 3 enzymes: exo-beta-1,4 glucanases, endo-beta-1,4 glucanases, and beta-1,4 glucosidases. These enzymes act sequentially in a synergetic system to bring about the breakdown of cellulose to generate a utilizable enzyme energy source in the form of glucose. Celluloses are generally divided into 4 major classes on the basis of their mode of action: exoglucanases, endoglucanases, beta-glucosidases, and cellulobiohydrolases. Microorganisms involved in cellulose degradation. A broad spectrum of celluloidic microorganisms, mainly fungi and bacteria, have been identified over the year. The structure and mode of action of the cellulosis produced by different microorganisms are also different. Cellulitic fungi. Fungi are among the most active agents of decomposition of organic matter in general and cellulosic substrate in particular. Cellulose producing fungi are widespread among fungi and include species from the ascomycetes, and this is a species name, basidiomycetes, with few anaerobic species. Among fungi, soft rot is the best known for producing celluloses, and among them, trichoderma is the best studied. Celluloidic bacteria. Celluloidic bacteria often produce celluloses in small amounts, and degradation of cellulose seems to take place by a cluster of multi-enzyme complexes. Most of the bacterial celluloidic enzymes are reported from Bacillus, Acinetobacter, Cellulomonas, and Clostridium. About 90 to 95% of all bacterial cellulose activity is observed under aerobic conditions by aerobic bacteria. However, the remaining 10% is degraded by a diverse group of bacteria under anaerobic conditions. Enzymes involved in the degradation of cellulose. We came for a main topic. Okay, the enzymes involved in the degradation of cellulose are groups of as celluloses. There are about 5 types of celluloses on the basis of the reactions they catalyzed. One is endogluconesis, second is exogluconesis, cellulobiosis, oxidative cellulosis. So endogluconeses are a group of endocellulose that cleave the cellulose molecule at the internal bonds of non-crystalline surface of the molecule. Endogluconeses randomly attack the cellulose chain and split beta-1,4 glucosidic linkages present within the molecule. Endogluconesis functions to reduce the length of the cellulose so that the fragments can be acted upon by other enzymes. So next is exogluconeses. Exogluconeses are a group of exocellulose that hydrolyze the reducing or non-reducing ends of the cellulose chains. The major products of the enzymatic actions are cellulobiosis, which are further hydrolyzed into monomeric units. Exogluconeses act on the smaller tetrasaccharides and disaccharides formed after the action of endogluconeses. Exogluconeses include both 1,4-beta-D gluconehydrolases liberating D-glucose from beta-glucan and cellodextrins, and 1,4-beta-D-glucan-cellobiohydrolases that liberate D-cellulobiose from beta-glucan in a processive manner. Next is cellobiosis. Cellobiosis are enzymes that act on a cellobiose units, disaccharides, trisaccharides, and tetrasaccharides to form monomeric units. Cellulobiosis are also called beta-glucosidases as they form individual glucose unit. Next is oxidative celluloses. Oxidative celluloses are enzymes that depolymerize cellulose into smaller units by radical reactions. Enzymes like cellobiose dehydrogenase catalyze the conversion of varied forms into cellobiosis so that it can be acted upon by cellobiosis. We have one more also, cellobiosis phosphorylases. Cellobiosis phosphorylases are similar to cellobiosis except that the hydrolysis of polymeric unit is brought about in the presence of phosphorus rather than water. So we have two forms. So in that, aerobic and anaerobic degradation of cellulose. So first one is aerobic degradation of cellulose. So we'll continue on this. Aerobic celluloses is performed by the synergetic action of 3 types of enzymatic activities. Endogluconeses or 1,4-beta-D-glucan, 4-glucanohydrolases, exogluconeses, and beta-glucosidases or beta-D glucoside-glucohydrolases, resulting in the release of D-glucose units from soluble cellodextrins and a variety of glycosides. Aerobic celluloses are produced in high concentration and also act as a sequential manner. Aerobic hydrolysis is fairly simple and occurs in sequential steps, each of the steps catalyzed by a different type of cellulose enzymes. Endogluconases attack more amorphous regions of cellulose fibers, forming sites for hexogluconases, which can then hydrolyze cellobiose unit from more crystalline regions of the fiber. Finally, beta glucosidase by hydrolyzing cellobiose result in the formation of monomeric glucose units. So next one is anaerobic degradation of celluloses. The mechanism by which celluloses form from anaerobic bacteria catalyze the depolymerization of crystalline cellulose is poorly defined. However, it is known that the mechanism is distinctly different from that of aerobic hydrolysis. The celluloses of most anaerobic microorganisms are organized into large multi-protein complexes called cellosomes. The cellosomes mediate a close neighborhood between cell and substrate and thus minimize diffusion losses of hydrolytic production. Factors affecting cellulose degradation. Cellulose degradation in soil or other is influenced by a number of factors, some of which are available minerals, temperatures, aeration, pH, organic matter, lignin. So I'll be explaining on this. First one is available minerals. The availability of nutrients and minerals affects the degradation of cellulose as these components are required for the production of biomolecules like celluloses and the other proteins. The increase in nutrients and minerals increasing the rate of cellulose degradation. Next is temperature. Cellulose degradation occurs within the temperature range of 0 degrees Celsius and 65 degrees Celsius as both psychrophilic and thermophilic organisms are capable of hydrolyzing cellulose. However, the degradation of cellulose is optimal at the mesophilic temperature range of 25 to 30 degrees Celsius. The third one is aeration. The availability of oxygen affects both the rate and the mechanism of hydrolysis as the enzyme involved and the mode of action differs in aerobic and anaerobic organisms. In the presence of oxygen, a sequential process of hydrolysis of cellulose in glucose occurs by their different groups of enzymes. Under anaerobic conditions, the cellulitic enzymes form a multi-enzyme complex which is comparatively slower. Next is pH. Cellulose degradation is slightly higher in acidic soil than alkaline or neutral soil. Under the acidic condition, fungi are the primary group of organisms involved in cellulose decomposition, whereas bacteria and actinomycetes act as a dominant cellulose decomposers in neutral to alkaline condition. The fifth one is organic matter. The presence of organic matter also increases the rate of cellulose degradation as much of the organic matter act as a substrate. However, if cellulose is the only component of the matter, the rate of hydrolysis decreases. The rate of degradation increases with the addition of a small amount of readily decomposable organic matter as it allows the growth of microorganisms. The sixth one is lignin. The presence of lignin decreases the rate of cellulose degradation. Lignin is closely related to cellulose, which affects the degradation of cellulose. So process, how it takes is, in a simple steps of cellulose degradations are, cellulose degradation occurs in three simple steps. One is hydrolysis by endogluconases, and hydrolysis by exogluconases, and hydrolysis by beta glucosidases. Hydrolysis by endogluconases, I'll be explaining. The first step in the degradation of cellulose is the action of endogluconases that randomly attack the cellulose fibrils. This step results in a decrease in the size of cellulose chains as it degrades the polymer into smaller fragments. The enzymes act internally at random points of polymer. The second one is hydrolysis by exogluconases. Exogluconases act on the smaller fragments, resulting in even smaller unit of tetrasaccharides or disaccharides. Exogluconases act on the reducing end of the fragments to form either dimeric units of cellulobiosis. The third one is hydrolysis by beta glucosidases. Beta glucosidases or cellobiose act on the dimeric unit of glucose of cellobiose to form monomeric unit glucose. This is the final step of cellulose degradation that result in the formation of pre-individual unit of the glucose molecule. So I have taken last one. This is the enzymatic degradation of cellulose chain via synergetic interaction of cellulose. This is the structure. So thank you all.